Amazing. Hello, everyone. My name's Krupa Tucker. I'm a second-year PhD student at the UCL Cancer Institute, but I'm also the Vice President of GBR. I hope you're all having an amazing summit. There's been some incredible discussions so far, and I can't wait to see what we all come up with. Our panel today, currently, is a panel of quantified self. So we'll be going over some topics on digital health, personalized medicine, and big data, some key issues in our industry currently. For our panel today, our chair was previously Tobias Silbersen, the head of the Health Tech Network, but unfortunately he can't be here today. Thankfully, Victoria has taken his place. As many of you know, Victoria is the executive director of the GAP Summit, but she's also a postdoctoral fellow at AstraZeneca. We also have Leanne Summers, who is the head of AI strategy for NHS England. We have Sir Harpal Kumar, who is the president of Biopharma and Europe at Grail. We have Rob Kotchie, who is the president of Real World Solutions at IQVIA. We also have Professor Sir Mark Caulfield, who's the vice principal at Queen Mary University. We then have Dave Michalovich, who is the SVP of Precision Medicine at BenevolentAI. So we'll be going through a really exciting discussion today, and stay tuned. Thank you.
Thank you, Krupa, for the lovely introduction. I think it would also be lovely for everybody to go around as well and say a little bit more about themselves and what they're doing. And then we can start as well the sort of panel, set the scene, and then go into the discussion. So I will pass on to Sir Harpal first.
Okay, lovely to be here. Hello, everyone. My name's Harpal Kumar. As you've just heard, I'm president of Grail for Europe and all of our biopharma business. Briefly, Grail is a company that's developing new methods of detecting cancers earlier and then following them up once they have been detected, using the power of next-gen sequencing, which we can talk about more if you're interested. Prior to that, long history in cancer, most of that time running Cancer Research UK.
My name's Leanne Summers, and I am nowhere near as distinguished as everyone here. But apparently you shouldn't be self-depreciating. I was part of the founding team for Global Biotech Revolution, though, so that's part of why I'm here today. A friend phoned me in. So, yeah, thanks.
Honesty is important.
Yeah, absolutely. No, but I do, so I lead on AI, strategic development work really within NHS England. So that's looking at how we sort of use the patient's data, you know, that we obviously deliver national health services here. Public trusts us to deliver their care. We need to be able to finance that. So there's kind of a bit of a trade-off in sort of data use, and obviously for biomedical science purposes as well. How can we accelerate the adoption of some of the technologies? So it's kind of like navigating that and trying to support with the digital transformation. So I've worked on things like the NHS app and NHS login, and a long history in sort of science communication and digital health startups, that sort of thing. Thanks.
Rob Kotchie, president of IQVIA, the human data science company. We're an organisation that supports life sciences, medtech and government throughout the world, basically to help molecules all the way from clinical research through into post-launch commitments. The business unit I run, Real World Solutions, uses data and technology to look at the safety, effectiveness and value of therapeutics when they're on the market as opposed for registration in clinical trials. Pleasure to be here.
I'll give you a flat clap. Hi there, folks. So my name's Dave Michalovich. I'm SVP of Precision Medicine at BenevolentAI. So as some of you may know, my company, or the company I work with, is involved in AI enhanced drug discovery. The particular team I lead, really interested in understanding complex disease at greater depth, going into phenotypes and traits, and then really move that information into a more molecular view using genetics and genomics data, and ultimately support our target discovery and also how we reposition our drugs back into the clinic as well. I'm a molecular and cell biologist by training, which I hang on desperately to in whatever I can do, as my team will know, and I've been in the industry for about 25 years now, working in pharma and biotech. So I've been in the record label and back in the band now, really. Anyway, I'll hand over to Sir Mark.
Hi, I'm Mark Caulfield. I'm Vice Principal for Health at Queen Mary, which means I run the Faculty of Medicine and Dentistry there. Previously, for 8 years, I was at Genomics England, most recently with Chris, who will be speaking later. And over that time, my mission was to deliver the 100,000 Genomes project and hopefully get a genomic medicine service, which we now have. But now what I'm focused on is about changing the healthy life expectancy of a woman in Tower Hamlets, which today is 58 years of age. She lives to be 82, but she lives for 24 years with ill health. So I'm going to apply all the things that I've learned, both at Genomics England and over my life, to addressing that health inequality for the remainder of my time. Thank you.
So really a diverse panel before us with a lot of different backgrounds and experiences. And really what we want to do today with this panel is talk about these opportunities that we have in data to really innovate and gather more biological data sets and genomics to understand and drive towards a future of personalized healthcare. But of course also discuss the challenges that come with it. You know, how do we use these data sets to create a fair and equitable solution for all of society, not just a privileged few. So I'd like to start by directing a question at Sir Mark. So Sir Mark, you have lots of experience with genomics via the 100,000 Genomes project, as well as with cancer registries, real-world data, and so on. So what impact of genomics on life science innovation have you seen in the past years, which you are most excited about?
Well, I guess taking the genome, which is about 3 billion letters, makes us the individual we are, gives us also, sadly, sometimes susceptibility to disease, and combining that with almost your entire health record can now liberate really major findings for people. And so as a result of, for example, the 100,000 Genomes project, where we work very closely with Harpal and CRUK, we actually found that we could use this to make diagnosis of about 25% of people. If you revisit their genetic makeup, because our knowledge changes all the time, this is a gift that keeps giving. And what you need to do is to combine it with a lot of clinical data. But if you do that, it can also be useful in changing cancer care. And that now is part of a genomic medicine service. So really, I would encourage everyone here, whenever they do a program of work, if it's about humans and human study, to follow the people for as long as possible. That's beyond the length of a trial like Rob does. So it's 26 weeks, follow them forever, because there will be long latent signals of benefit and long latent signals of harm. So that's how I'm looking to apply it.
Thanks very much. And Dave, you also have a great perspective on life science innovation, both from your current work at BenevolentAI and also at GSK, where you worked before. So with the topic of genomics, can you maybe share a bit of your perspective about omics as well?
Sure.
With regards to transcriptomics, you know, what's the role of these other?
Sure, yeah, sure. So I've kind of been privileged to be in the industry for about 25 years now, and I joined at the kind of advent of human genome sequencing, and there's this massive hope that once we sequence the human genome, we would find all the drug targets, and we got very excited. We were throwing G-protein coupled receptors over our shoulder and new kinases as we mined through this data, and then we got this bottleneck really, and what was really missing for the industry was the biology, and it's taken quite a number of years really for the technologies to really catch up to allow us to understand the functional impact of genomics. And I think some things have changed really over the last 15 years. So industry did quite a bit of sort of introspection, navel gazing around how its portfolio was doing. And some of the findings for that was actually that a lot of drugs were failing at phase 2 once you got into efficacy. And really some of the drivers for this wasn't safety, it was actually the efficacy aspect. And there's a realization that we weren't really understanding the patient well enough and selecting the right drug target. Further analysis of portfolios came up with some key findings of which data types are really important here. So work by AstraZeneca, by Matt Nelson and team at GSK, publications by AbbVie, and a recent one by Open Targets, show that if you have genetic validation on your target for the disease you're trying to improve or help, that you're 2 to 8-fold more likely to be successful in the clinic, ultimately when you get the thing approved. And the strength of genetic finding really improves that. So that was a real driver in industry to start looking at genetics at scale. That was coupled with the growth of next-generation sequencing and the ability to generate far much more genomic data. and also now the ability to understand the causal effect of genetic variants. So combining human genetics with epigenetic data, EQTL data, moving to knocking out genes or knocking out variants even at scale, genome-wide scale, using single-cell CRISPR type of technologies. That's been a really exciting innovation. I think furthermore, there's the application of metabolomics, proteomics, I think more in the biomarker space, really, but you're seeing some great papers where you're combining these data sets together, genetics, metabolomics, genetics, proteomics. The whole world of single cell, which is just blowing me away at the moment, really. So I think we're seeing this depth of data, depth of biology, which is coming from the omics field now. And this just means there's so much data to handle, and this is why we've got to be thinking about AI.
Thanks. And what are the challenges that you've seen in terms of integrating these data sets? This is kind of an open question to everybody if you've worked on them. For example, Rob, you've also worked on the electronic health records and real-world evidence data. So perhaps you can also comment on that.
Yeah, I mean, I think the first of all is data everywhere, not a drop to drink. Having the data is fine, but we need to understand the provenance of where it came from. You touched on a very important point, which is linkage. You need to be able to link the genomics data through with the clinical data to new aspects, social determinants of health, connected devices. But having the data alone, we also have to be honest about how it was captured and how it's used. Now, for research, there's huge amounts of data and we cannot just ignore it, but we also have to be very honest that the data used to capture in an electronic medical record is used to help the physician manage his or her interaction with myself as a patient. It was never designed for research. So I think things around standards, around not only the data types, the coding, but also the methods are very, very important. But the ability to be able to link across all of these domains is opening, I think, the world up. And I remember when Sir Mark and I started with some things IQVIA and Genomics England had done together, and almost the geneticist was coming at it, wanted to do all the analysis on the genome and then look at the clinical sequelae. The clinician real-world guys wanted to segment the disease and then look at the gene. So the ability to cross those worlds, I think, is becoming very important.
I think also, there's the data behind the real-world evidence, with the real-world evidence, and also data that's collected, for example, from UK Biobank. And perhaps Dave, you might like to comment on the use of biobanks, maybe share a bit of your experience there.
Sure. I think Sir Mark hit the nail on the head earlier. It's really being able to access resources where you've got deep clinical phenotyping. So UK Biobank is a fantastic example, but actually one of the very few, really, where you've got hospital records, you've got primary healthcare record, you've got blood biomarkers, we're getting imaging data, we're going to have 60,000 people with single-cell blood transcriptomics coming through, metabolomics, where you've got this depth of data. It is actually really rare to be able to find those resources, really. And quite often you've got a disease you're working on, you really want to get to those gut biopsies or those lung bronchial alveolar lavages, etc. And it's really hard to find sources for this. People, people aren't really aware of really what they have in the freezer or not, or how hard these things, so you tend to get these ambitions to promise you, 50 subjects or something like that, and it really doesn't play through. So get access to data is really hard. Having the deep phenotyping is so important for what we do. And I think there are some brilliant resources, some fantastic results coming out of genetic analysis and other analysis of UK Biobank, new genes associated with diseases, which are really important. But we've got a long way to go to really understand the human body, I think.
And what do you think maybe is something we can do to increase the access to these data sets to create that deep clinical phenotyping you're talking about?
I think we need to engage with the public. So we've launched this year Data Saves Lives, which is a data strategy specifically talking about sort of the uses of health data with the population. I know that Genomics England are doing work on health equity, sort of the uses of data, and having discussions as well. If you've taken tissue, who does that belong to? And have we ever actually had that discussion with the public? Because if you start getting into that, it's kind of a can of worms that it might be better to get into now than much sort of much further downstream. So had some really good conversations with some of the, members of the audience, the work that they're doing. It's sort of like, where are you actually thinking about the ethics in the science as well, so that we can translate that science into something that's really tangible. And we've had this opportunity with the pandemic. I mean, obviously it's been terrible, it's not really an opportunity, but people are engaging, with the stats and this sort of thing now. So how do we sort of monopolize on that and keep that dialogue going? And what you were talking about, sort of like data entry as well. Where are we engaging with clinicians and frontline practitioners who actually have quite a crappy user experience in terms of entering the data?
Yeah.
So that we actually get the high, better quality data that we need. There's things that we can use as well. Sort of, we've got some tools that we've funded through their AI and Health and Care Award that are looking at sort of, as people enter. It's not just the projects that we've funded, Microsoft, other providers have these technologies as well, where, voice to language conversion is sort of typing, it's typing up the notes, it's then auto-tagging it. It's not there, it's still, it's not going to completely automatically do everything, but the point is it's sort of emerging. So how do we improve that user experience as well to get that better quality data that will support your science actually being better?
I think the point you made is very important. If you think about the data in the health system, just the health system, only 20% of it is probably structured and coded. Yeah. A lot of the other thing is unstructured data in the free text and notes. So technologies to be able to structure that data, understand the accuracy and failure of some of that, I think is going to be very, very important.
So I suppose this is an obvious question now, but what's the role of machine learning, artificial intelligence in all of this?
I think it's going to help to accelerate sort of the triangulation of some of these data sets. This is what we're really interested in. We're seeing applications sort of with imaging in X-rays, MRIs, this sort of thing. But I actually think that the biomedical application of it, where it's being used in pathology, laboratories, that sort of thing, and anything that you're doing, like if you can do a computational course, just do it now, because you're going to need this sort of as you're progressing with your career. Sort of what can we do? Lots of different things when we're triangulating these data sets.
I think it's important just to recognise, and of course this is something that's widely said, but machine learning is a tool. It's an approach, and what's really critical is understanding the question you're trying to answer, and the power of these tools is now immense. The quantities of data that we're dealing with are going up exponentially and will continue to do so for at least the next couple of decades, I would think. And so we're going to need these approaches, but actually it isn't just a case of saying, well, let's take a machine learning approach. It's a question of saying, what is the question I'm trying to address? What's the data set I need to be able to address that question? And then how can I bring these technologies and approaches to bear on answering that question?
I would layer on in terms of, there's a difference between using machine learning for, say, hypothesis generation versus hypothesis testing. The actual clinical feasibility, understanding of the mechanics, the mechanisms of action, and the ability to replicate something. So you run analysis in one data set over here, and then you test it on another set over here. Things of that nature, so you see consistency as we draw in the I think it makes the interpretation probably more difficult, because we've now got a lot of findings, right? I mean, classical statistics, right? We run 100 tests, 5 can be positive this way, 5 positive that way, right? So I think the interpretation and the science of that, and what we declare ex ante versus ex post, is going to be important as the field evolves as well.
Yeah. I think your point around having training data sets and validation is key for the ML technologies. We have some great ideas and applications of these technologies, but often the question is, well, actually, how are you going to validate this? And when we talk about drug discovery, often that validation isn't really until you're in the clinic, right, ultimately. So where do your surrogates come from? We're talking recently about looking at variant to gene mapping and using machine learning tools to do this. And there are a number of applications out there already. DeepMind's got one. There's a number of other ones out there. So how do you validate that? And ultimately, you probably need to do the CRISPR experiment in a cell, knock out that variant and see which genes are changing. And then you've got to do it in the right cell. It's not just any cell. And so these become quite massive experiments to really be thinking about leveraging. So you've got to be thinking about carefully where your training data sets are and how you're going to really validate these things.
I think that's a really important point, that once you have identified the hit, you have to do all these tests early on to make sure that you are getting the right hit, and it's not just, a false positive that you figure out way down the line in clinical trials phase 3 that it's not, that it's a dud. I think also, Sir Harpal, you raised a really important point about, making sure you have the data set to answer the question that you want to ask. And, I find from my personal experience, sort of doing postdoc research in industry, it's quite interesting because much of the time the data sets in industry aren't based from a grant, for example. So you don't collect the data, you don't ask the question and then collect the data. You have the data and then you think, this question is really interesting. So what data do we have? And often we collate that data set retrospectively. I want to ask if this is a sort of something that you see in your own fields and what your thoughts are around this.
I mean, I think what you're describing is true across all of biomedicine, but it's probably true across all fields of science, actually, which is, you have to start somewhere, and you typically start with what do I have available to me, and Rob just described the difference between hypothesis generation and and testing, and I think it's completely fine to start with the data sets you have, but what's critical then is to say, okay, that's a hypothesis I've generated. It's not the evidence. How do I then collect the prospective data I need to be able to have confidence in what I'm seeing, and that then does have to be collected rigorously with the question in mind that you're trying to address, so that what you don't end up with is a biased result.
I would agree with that. I think there's a unique opportunity for the United Kingdom because of a single payer system. We could aggregate our data en masse. We could, by, as you rightly suggested, getting people, patients, public involved at the outset to shape that, we could create a core data set. If you then consent those people to engage in longitudinal life course follow-up, you can do what Harpal's just described, which is recall the right people to avoid a biased result and retest your hypothesis. And you can also use that as effectively an opportunity to create a funnel for appropriate clinical trials, because clinical trials are so expensive to do. And what we need to be able to do is to make that more affordable, particularly for rarer diseases. Drugs often won't be adopted by payers for that reason. And we now have very excellent cancer therapies or immunotherapies are also expensive. Can we use a combination of the genome with their clinical data to get the right drug to the right patient first time? And by the way, also avoid harm downstream. So we need to take a proactive, prospective perspective on this, on aggregating a standardized data set, but using that framework to recall to answer the more detailed question, which several people have said, and that's the deeper phenotyping. So you can't do deeper phenotyping for everyone, but what you really need to know is where are the people who would most potentially benefit from this innovation, and those are the ones you need to recall. And that's why moving to early prevention and detection is really important for the health system. But we can only do that effectively if we're going to do things at scale, because we can't find the people otherwise that we need to. So one example is what Harpal's doing now with cell-free DNA, which is using DNA released from tumours. You might want to say a bit about it and how it helps us to detect.
Sure, but I don't want to disrupt the flow of the questions. So
Go on, Harpal.
Well, I mean, I think Mark's alluded to it. Which is. If you look at where we've applied genomics over the last 10 or 20 years, it's really been in developing and optimizing therapeutics and using the knowledge that we've gained to optimize treatment for patients, all of which is fantastically important and critical, just to be absolutely clear. But if we're going to have sustainable health systems in the future, we can't just be treating people with very late-stage disease and chronic ill health, which is sort of where we are right across the world now with the health systems and the approaches we have. So we have to take an approach which enables us to get ahead of disease, and that's in two respects really. One is, can we prevent more people with illness can we prevent people getting illness in the first place? But secondly, can we detect it earlier? And in the context of cancer, which I'll use as an example, but this applies across other diseases as well, we know that the vast majority of people who die from cancer die because we're detecting those cancers very late. And that's because very often symptoms don't appear until quite late in the disease course. And so if we're going to make a really big shift in cancer, then the way to do that is to be able to detect it before symptoms appear, which is a really tricky thing to do. But actually, what we're increasingly understanding is that through the release of DNA by tumour cells into the bloodstream and into other fluids, indeed, that we now have levels of sensitivity that enable us to find those very, very, very small quantities of that circulating DNA in amongst all the noise that there is in the blood, and then we can use that information to determine whether an individual does or doesn't have cancer. And the approaches now that are emerging, including those that we're working on at Grail, look extraordinarily promising in terms of being able to do this. And that opens up a whole wealth of new opportunity and a whole wealth of new science that needs to be done. We know, okay, so we can find these cancers earlier. So what does that mean for therapeutics? It means that we have the opportunity to start applying therapeutics earlier in the disease course, which is actually when they're more likely to be more effective. It means that we have the opportunity to really understand what early biology looks like in different diseases. We know nothing about the early biology of cancers yet. Despite all the work that we've done over decades, we know very little, and even less in areas like Alzheimer's disease or many cardiovascular diseases and so on. So the power of these technologies to really make a step change in our understanding, but also then in how we apply that understanding to shift from what is euphemistically called a national illness service into what is really a national health service. That has to be the way forward.
Or maybe even a wellness service.
Or a wellness.
That would be really
Or a wellness
nice.
service.
My reservations, so I think that this is brilliant, and in terms of testing, we've seen, MRI scans. If you want to go and get an MRI scan, you have to literally go to the scanner. There has to be a trained person there to put you in the scanner, a trained person there to interpret it, that sort of thing. So if we can get sort of spit in a cup tests, this is phenomenal. It completely changes how we deliver care. And what were we all doing the last couple of years? Spitting in a cup all the time, or putting a swab down the back of our throats. So we know that we can deliver healthcare like that. What I'm concerned about is when will we actually find the opportunity to find it earlier? We're going to have to sort of put in some, I'm 35, let me go and spit in the cup. Or maybe when someone's pregnant, or when there's these life changes that mean that there's a reason that you may be going into the healthcare setting, and that's a bit more routine testing, which might seem a bit big brother, which is fine, if people are happy that that's fine and they understand the benefits of some of this sort of work. So we're really excited. We're looking at what we're calling sort of, you've said it already, pathway optimization. So where can we work with teams within NHS England that are looking at, cancer or child health, whatever, and just make sure that we're mapping the service so that we can see opportunities for where maybe some technology like a testing. So for me, a genomic test is the same as a piece of machine learning in many respects, because it's just technology. So you're applying technology to that pathway of care. But what are those sweet spots that we can do that in? We're just as the NHS England sort of it's evolving, NHS Digital is kind of coming into one space. So that idea of that big sort of data repository, it won't be a repository, it's probably going to be federated, who knows, But the idea that the data will be accessible and there'll be access controls and what we can do with it. We are quite early days on the cusp of that, and the SDE policy, secure data environment policy, for the delivery of care, will accelerate, and then research and development opportunities will come off the back of that, I think.
Yeah. I mean, what you're describing is of course critically important. It's all very well having these technologies, but understanding how you're going to apply them, who you're going to apply them with, is a really important consideration, as is the conduct of really well conducted, really rigorously conducted clinical trials, because you need to see how these things work in practice. But there are in certain indications, and cancer is certainly one of them, it's somewhat easier to define some populations than it is in others. But equally, the technologies that we now have are going to help us to better target those interventions in the future. So, we're beginning to see, and we're beginning to understand at a much deeper level than we have done previously, who has a genetic predisposition to certain diseases, and whether through whether through work that's being done in various academic groups looking at polygenic risk scores, whether through new initiatives like Our Future Health, which is being established, we will have a much better understanding in the future as to, well, look, this individual, just because they're 65, doesn't necessarily mean that they're a really high risk of breast cancer. We need to modify that understanding with what we're able to deduce from both their clinical record, but also what the genetics will tell us about that individual.
But to build on that, if we move away from cancer for a second and we think about sort of the germline sequencing and the predisposition, you spoke about when to do the testing. The most cost-effective time to do the testing, presumably, is at a newborn, right? I mean, in the future, shouldn't we have our whole genome sequence appended to our medical record and every clinical decision pings off it? Is that where we should go here if we think about, what actually creates the most value for society over time?
And we have done public and patient consultation on this around a limited newborn program, and Chris will probably talk about that, so I won't say too much, except to say, if in the future it is impossible that we would be able to detect certain rare diseases early, and those might present before your 5th birthday, and that baby might be seriously disabled, and there was an intervention that you knew worked. That would be an opportunity some parents would wish to take, and understanding that's important. But of course, once you have that genome, it is your blueprint for your life. It doesn't change very much. It's largely constant. You can revisit it and interrogate it with agreement and consent of individuals. So I do think in the future we will end with much earlier, more comprehensive. It's not a screening using that approach, but it's not affordable now. We haven't proven it yet. But I think you would want to give people the surest start in life. And then to be able to say to them, actually, if you took this and this action, maybe to your wellness point, which I think is a very important one, you might be able to have a healthier life. Now, I think, the problem for us is when we get to our 40s and 50s, we've established these processes. Now, we can still do something about them. We still have good treatments, and we can still detect our risk, but it's harder to sort it out. And that's to Harpal's very important point of not running a national disease service. And the other thing is we had until recently no good biomarkers for cancer, really. And now we've got to take that chance because sadly 1 or 2 of us will probably have cancer at some point. And we can show from cardiovascular disease, if all you do is aggregate big data and you intervene on the practices in general practice where blood pressure control and cholesterol control is not so well done. In East London, we've saved 300 lives and 20 million to the health economy. So there is opportunity to generate really big wins. Cardiovascular disease has got good biomarkers. Cancer's becoming good biomarker territory too.
Well, one approach I like the idea of is actually looking at the sort of temporal view of disease.
Yes.
And looking at the genetic signals for disease progression. So quite often when we look at, say, asthma genetics, yes, you'll get a number of signals, but they're not necessarily the genes that are actually going to be involved in actually whether you're going to progress or severity, etc. Now we have these deeper phenotype cohorts with temporal data. We can start looking at the genetic signals which say how you're going to progress in disease. And I think those progression genes are going to be really interesting because they're probably more likely to be our drug targets for modifying disease. When I worked in COPD, a number of genetic signals in COPD were actually probably playing out in utero, around sort of lung development, etc. And then if you impact that with smoking, etc., then it probably plays out with COPD. But as a drug target space, it was very limited, really. So I think that those sort of considerations we should bring on board.
I know we're talking a lot about personalised medicine and genomics and omics, but there are other data sets. If we think about, I don't know how many people in the room carry a smartphone, probably 100% of you. If you think about predicting things like Alzheimer's or early disease, right? How many keystrokes do you make failure per day? How does that change over time? The role of wearables and connected devices in either early disease detection, I think, is going to be critical. But then longer term, is that also going to be the outcome measure? So we used to run in clinical trials, hard endpoints, cardiovascular event, death, et cetera. You've got a lot of these patient-reported outcomes, these sort of softer measures around function and feeling, which are actively reported. So you mentioned COPD, the St. George's Respiratory Questionnaire, fill it in. But we're now getting to the point where a lot of those wearables can actually detect that data passively. And I think that's going to be important for two reasons. It's going to be important for how we help for those that consent, again, for early detection of disease. But eventually, if we can validate those, the drug gets approved on the primary endpoint of COPD exacerbations, St. George's Respiratory Questionnaire. We validate the secondary one. Suddenly that changes how the patient and the health system interact, because we can monitor that constantly. So there's a lot of talk, which is right, around early detection, around those different pieces. But I do wonder, if the role of those devices and other sort of attributes, social determinants of health, are going to have as big an impact in healthcare deployment than genomics in the interim. And obviously those two need to come together.
There's undoubtedly really good information in those data sets. The question is whether we can use them to robustly define endpoints that regulators will accept. And regulators in this industry are extremely important to anything we want to do. But I mean, just to give you a couple of illustrations, again, from the sort of cancer world, is, we know through relatively small studies, but nevertheless, that a couple of things that happen prior to someone being diagnosed with cancer. If people are going to be diagnosed with a brain cancer, you see in the months before they're diagnosed an increased pattern of buying headache pills from their pharmacy, because guess what? The most common symptom of brain cancer is persistent headache. You see that, prior to people being diagnosed with other types of cancer, increased levels of Google searches for different types of symptoms. So we know this information exists in other data sets, but how you disaggregate it and how you make it a robust marker for whether it's detection, whether it's disease monitoring, whatever it is we're trying to do, is still a challenge, I think.
I do think, though, the point Mark was making earlier about the standards, data standards, the curation of data standards. We've got the so the NHS app, NHS login, launched, let's say, low-key, 2018. We had about 1 million downloads. Lots of patients were sort of changing their prescriptions, that sort of thing, booking appointments, but not very many downloads. And then, obviously, when you weren't allowed to travel, we got 22 million downloads. So now, that's actually, it's a little bit under half of the population of England have logged into the NHS app. Now, the important thing for me is the actual login functionality, because that login provides us with a portal through which we've got sort of a permissions gateway. So we could say, do you want us to track your headache pills, or these kind of things, and we could probably get it down to quite a granular level. I'm talking sort of hypothetically, we're in a sort of strategy room here or whatever. This is not policy, but it should be. But yeah, we actually, 22 million people have already logged in. That's huge. Why are we not looking at that more?
Perhaps we should let you ask us some questions now.
I've been letting this go because you've been answering all of them without my having to ask them, so this is perfect. I think we're almost to the Q&A time, so I think one question I'd like to ask all of you is, what's the innovation that you're most excited about, and what's the application that you're also most excited about for the future? Asked a tough one there.
Well, I mean, I think I've already answered that from my perspective. I mean, I think we have the ability to totally transform how we think about really serious conditions that affect, the vast majority of humankind. If we could shift from detecting, half of cancers at late stage to being able to detect them pre-symptomatically, we transform the condition entirely. Completely, for all time, globally, and that is the vision that we're on, and this doesn't just apply to cancer. This absolutely applies to other conditions as well. We detect them far too late at the moment, which is why we then have to invoke quite intensive, quite expensive therapies, which work and are great, but they're not the answer to the future of health. So that's where we have to go.
I agree with that. I would add into that, I think we should detect disease early at the extremes and the middle of life. So we should have life course health, and we should use a multimodal data accrued from the health system routinely, which we also, by the way, use to manage real-time management information for the system. And that if we do that and we layer additional omics depth on that, we could be able to transform rare disease, I think cancer and cardiovascular disease. And if you get cancer and cardiovascular disease, you've got the biggest reason any of us here will die.
It's hard to beat those two, I have to say.
That's why I got mine
I dream of those. But being a molecular and cell biologist, the stuff that actually, other things that excite me, I think the single cell world is amazing at the moment. And the single cell combined with spatial transcriptomics, and we're able to refine our view of effectively which cells are talking to which cells in a tissue, particularly a tumour. Getting these sort of molecular barcodes, thinking about tissues and disease as cell-to-cell interaction networks rather than protein-protein interaction networks, really exciting. Those interfaces are going to be where new therapies are going to be found, really. So I'm very excited about that as a molecular and cell biologist, but as a more kind of a precision medicine, I would share the other ambition.
I'm going to go policy wonk, completely different. I'm really excited about sort of like what you've been presenting today, the intersection of different things. So like looking at the climate, looking at diet, looking at, you know, our ecology, that sort of thing, the soil structure, things like that in terms of wellness sort of side of things. And I don't think we're doing enough to sort of look at the science. So like my kind of look to the future is have maybe a few more of you guys interested in policy work so that you can act as a translator, because I feel like that's sort of role that I have to play, where it's sort of this hardcore science over here, like this stuff is amazing. I wish I'd had more time to spend in it, but I'm spending a lot of my time just even explaining, just even trying to get that simplified to some of my sort of programmatic colleagues who are wonderful and they, you know, deliver health service, but we can't get that change. So it's kind of a boring one, but I think it's really important. It's not going to change otherwise.
I think I see where you're coming from, because I think one thing we've sort of touched upon is, but not really said explicitly, is that we need people on board with all of these different technologies.
Yes.
We're all assuming right now that everybody will be happy with these technologies, that they're going to improve healthcare, but really for different groups of people that's not the case. And of course one of the challenges is how do you talk to different populations, different peoples, and explain how this technology might benefit them in the future for healthcare? Rob, do you have any?
Yeah, I agree on the early detection and prevention, I think, is key. The other area that I think we're going to see, hopefully, a huge amount of progress is, we've spoken about cancer and other things, but in CNS, in areas where the endpoints are more difficult and the ability to actually capture what patients value in some of those conditions, I think, is going to become increasingly important. So that's an area that I'm very excited about.
But I also want to pick up on something that you were just talking about there, which is you know, acceptability of new innovation is a really critical thing. But so is the whole area of health equity. Yeah. And what we've seen, unfortunately, over the last few decades is an increasing ability of those with resources to be able to access the highest quality care, whether it's in this country or whether it's in the US or whether it's anywhere else in the world, frankly, but actually an increasing proportion of people who can't access these new approaches, these new technologies, some of which are actually very affordable, but all of the sort of practical issues around access, around availability, around affordability, are areas that are going to be critical for us to address in the future. And it isn't just about the technology, it really is about understanding what is it that makes someone decide to come to an appointment or not come to an appointment. Yeah. What is it about their ability to even think about that in the context of going to work, looking after their kids, or worrying about, you know, the cost of the bus fare or whatever else it might be. And what is it that stops clinicians using the new technologies? You know, if I've only got 4 minutes to spend with a patient, I don't have time to talk them through all of the implications of a genetic analysis. So how do we find new solutions to some of those sorts of things? So the whole issue of access and equity, I think, is going to be a huge challenge for us to address in the future.
I'm not sure I personally don't have the answer to this, but I think we can open the floor for Q&A and some more discussion.
No interest.
And a lot of interest.
By the way.
Thank you very much. I'll try to keep the question quick so we can get to most people here. I was thinking about the multimodal nature of the data that you're describing and the role of AI in it. And I think the medical apparatus has a very defined, probably because there were some high-profile mishaps in informed consent throughout the years. And then you start to think about things like wearables or like interactions with technology where it's governed more by contract law and user license agreement. And so at what juncture, like I haven't read most end user license agreements I've signed, right? At what point does, the synthesis of these different things start to border on uninformed consent and erode some, faith in the medical apparatus? The idea of having your genome sequenced at birth is all well and good, but a baby's parents can consent for things as a child, but that's, as you said, forever, and you can't take that back.
I think the opportunity is now for us to have that conversation. Like, we have to have that conversation now. It's critical, because otherwise, we're just going to completely erode trust. We've done it time and time again, accidentally, like, sort of care.data. We keep sort of making own goals, so we have to start having those conversations. And I do think, centrally, we're a bit nervous about saying things like, we'll use your quantified self data, because, actually we don't want to do that unless you want us to do that for you, and we've got that, sort of trust established. So that's my thoughts anyway.
I was just going to layer on, throw something that's even more horrible into that debate, is the nature of consent is very important about opt-in, opt-out, explicit, specific, etc. But let's just take the genome, for example, right? If I consent to use my genome, I'm an identical twin, I've just disclosed all the information on my brother. So to me, it becomes a societal debate. It's a herd problem. We have to have the societal discussion. People should have the right to opt out, the right to be forgotten. But I think it needs to have that broader platform. The best paper I've read on this is called The Rule of Easy Rescue. I can't remember who it was written by, sorry. And they basically say, look, we need linked medical records to run the health system. If done appropriately, you can gain all of these additional benefits for research without sacrificing the individual citizen's liberty. But that's how fundamental this debate is, right? Collective value for society versus individual citizen's liberty, which is why it's also very political, inflammatory as well.
I think one of the answers is that to your end-user license agreement, I think you don't rely on that. You rely on securing consent that's broad but describes the type of things that you'll do. Our experience when we initiated consent in Genomics England in 2013-14 was that we as clinicians thought there were all these things the patients wanted to know. They went through that consent material and said, no, we don't need to know this because we know this already. No, don't need to know that. And actually they shortened it considerably. They said, this is what we need to know. Otherwise, this looks like an end-user license agreement, and I don't want to sign that, or I don't want to sign an insurance agreement. So you have to create an appropriately balanced consent material set that allows you to be optimistic that you've gained consent, confident that there's an shared understanding of what's going to happen, and then you can operate. To your point about children, in childhood, our mum and dad do consent for us up to 16. But there is no way you can work on the data after that age. And as soon as they are judged to have a competence where they could understand those issues, you could proceed to consent them. So even if you donate your genome, your parents do it as a child, you still have say in what happens. So it's not a foregone conclusion. And I wouldn't support that, because every one of us is an autonomous human being.
And we change our mind.
And we change our mind, yeah, especially when we're about 16.
I'm back again.
Just choose someone.
Hi, so I basically, this is probably like a naive question, but regarding cell-free DNA being used as a tool to detect cancers early, like pre-symptomatic. I was just wondering how you would kind of choose people who kind of, take on those diagnostic tests, because when I've worked on cell-free DNA briefly, it was in the context of trying to understand whether we can use it as a marker to follow on treatment responses. But I think the concept of using cell-free DNA in pre-symptomatic patients just seems really interesting, and I was just wondering how you would pick on those people.
Yeah, so there's actually sort of three different sets of use cases. One is the one that you described. So can you use circulating DNA to follow patients once they have been diagnosed, when they're on treatment, and so on. So I won't talk about that. But there are two other contexts, actually. So one is the one that I was alluding to earlier, which is you define a population at risk, which in the case of cancer, your risk increases very rapidly from the age of about 50. So we know, which is why most screening programs start from the age of about 50. So we know that your risk increases very rapidly from that point onwards. So that's a good age to start, and you run trials, and that's what we're doing. And we're running trials to see, just how much of a difference would it make if we screened people who are otherwise healthy from the age of 50 to about 80 for the presence of cancer, which they would have no idea about. There is another use case as well, which is also worth describing, which is we know that, again, and I'll talk about cancer, but it applies to other conditions as well. The vast majority of people are detected when they go and present to their GP with symptoms. But actually, the symptoms are often very non-specific. It might be that the individual's lost some weight, or they've got some abdominal pain, or they've got some anemia, or whatever. And in the vast majority of cases, those are not cancers. But the GP doesn't really know what to do with those individuals. So we can also use the detection of DNA to be able to help the GP differentiate between a condition that's really likely to be serious and one that's likely to be less serious. Doesn't mean that the symptom doesn't exist, but it might be that it's something much less worrisome. And we can help to, if you like, triage those individuals that really need urgent investigation vis-a-vis those who don't.
Yeah. Hi, my name is Emil. I'm from the Bioinnovation Institute in Denmark. So we actually had a project in our group where we tried to investigate the bridging between translational science and pharmaceutical industry. And I would like to ask you, how do you bridge the gap between creating new pharmaceuticals, because they take up to 20 years and can cost up to $2 billion US to produce. Yeah.
That's one for you, Dave.
I think so. So I think it's, that's a tricky one. So I think the introspection the industry has carried out is actually just trying to say, what data do we really need to help us predict success early on? So when I think historically targets have been selected. Sometimes somebody's just read something in Nature and go, hey, that's really interesting, let's get going, or bouncing off previous pharmacology. I think now we're trying to get patient-level data, so genomic and genetic evidence for that gene being involved in the disease mechanism, trying to get that at scale so that you really believe that finding, and then move your drug discovery forward on that. Along that journey, the other important piece of data that we really need is to be able to translate the modulation of that target in our cell assays, in our animal model assays, through to clinical biomarkers, really. And if you've got that daisy chain of information from experimental work into the clinic, that also is a great predictor of success. And that came out of a lot of the AstraZeneca's analysis of their portfolio. So this is where we need to invest. This is where we need to understand the data in our basically prediction of likelihood of success. So when we're starting to assess our drug discovery portfolios and want to put our investments as pharmaceutical companies or biotech companies of which we think we're going to be successful, it's looking at that type of data for our probability of getting to that clinical point.
And we need to use the lessons from the pandemic, which are that we can actually accelerate the investigation of these therapies and bring them to life. And who'd have predicted within a year, 18 months, we'd have vaccines that we could use to tackle this virus that might normally have taken us 10 years. So we must never, ever go back to the way we were.
Yeah. It's not a data answer, but it relates to questions that were asked in previous sessions. Multidisciplinary teams and multidisciplinary individuals are critical for all of the innovations. So as people move, we kind of, at IQVIA call it kind of squiggly careers. People end up doing a whole bunch of different things, but a lot of the innovation in research comes when fields collide with each other. So, which is why it's great to have people from different domains. And it touches also the environmental point that was made earlier. So that would just be my other parting comment as you think about how do we continue that innovation is moving people across disciplines and fields and bringing people in from different areas, I think is key.
Yeah, absolutely.
Hi, my name is Deborah. I'm from Brazil, and I work in a medical device company that is using electric fields to treat cancer. And I actually work in the clinical trials area. So by now, we all know the impact that new technologies can have on reducing the time in clinical trials. But I was wondering, and for any one of you who want to answer, what is like a new technology or a new process that you personally wish existed that would make your life easier? Not necessarily what you think would be next, but what you really wish, and please be creative as you want, not necessarily something that is tangible, but...
I have very much a process one, which is the time taken to start up clinical research sites, even research sites that work all the time, and the time taken to get through sometimes ethics, which should be appropriately done. It's mind-boggling how long some of those processes take. And so if I had a magic wand, it would be simplifying in an appropriate way that doesn't, circumvent the safeguards in place, how we can set that up a lot quicker. And as part of the pandemic, centralized ethics in the UK and various other things to be able to move quicker in clinical research. So not very exciting, but I think removing the process to mean that we can actually engage quicker, I think would be my sort of wish.
I'll add on a comment or two, but I agree with Rob. It's largely about process, and so I'll give you an example of something that's worked really well and something that I wish worked better. So we've just finished enrolling our main trial in the UK. We enrolled 143,000 people in 10 months. How were we able to do that? By working with the central NHS digital system to be able to invite eligible participants from their database. Not something Grail was doing. We worked with the NHS to construct something that didn't exist previously. It was designed for COVID studies, but actually it needed a lot further development. But as was proved during the pandemic, you can actually do things in a different way. And so we were able to invite the right people and get them enrolled really, really quickly. What would have been much better, though, we talked about it earlier, is if we had better data linkage, because what we'd like to have been able to do is to exclude certain people from being invited, because we knew they wouldn't be eligible, but because the data exists in different systems, we couldn't avoid inviting people who otherwise would be ineligible. And all of these things take time. And for as long as every one of those steps takes longer, it makes the whole process of clinical research take longer. And so the actual conduct of the intervention is often very quick. It's all the process around it that tends to take the time. And so that's where I think we need to do most of the innovation in clinical research.
I'd get Treasury to look at how they fund program business cases. They don't really look at some of their, we talk about outcomes, but what they measure us on is sometimes activity and benefits. And you're like, this is just making that process that much harder. We could actually be, changing things here. But anyway.
I think we're just about out of time. So thank you everyone again for such an insightful discussion.
Thank you. Thank you.